Hello everyone, and welcome to B3.1, which is gas exchange in IB biology. Let's get started. So, gas exchange is performed by all organisms, so it is a vital function. It requires a large surface area. Why? Because it is a very slow process. But large organisms have a small surface area to volume ratio, right? So if you think about a human, right, the surface area of the skin is very small compared to our volume. And also there's a very large distance between our center and the exterior. Therefore, you need adaptations, right? You basically need a surface that is much larger than the outer surface of your body. And that is the alveoli in the lungs in the case of humans. Therefore, surfaces for gas exchange need to be large, as we just said, but they also need to be permeable. That means that CO2 and oxygen need to be able to diffuse freely. They need to be moist, so that means that gases have to be able to dissolve. That's what moistness allows for. And then they have to be thin, so gases must be able to diffuse through a very short distance, because otherwise it would make the process very, very inefficient. So these are the four main things to remember about surfaces for gas exchange. Now, gas exchange happens only if there are concentration gradients. That's very important to understand. So these must be maintained. So how's that done? Well, that's done through continuous blood flow. That happens through dense networks of blood vessels, which carry a low concentration of oxygen and a high concentration of carbon dioxide. Why? Due to aerobic respiration. So cells use up oxygen and then excrete CO2. So that means that in the blood, outside the alveoli, there's going to be a high concentration of CO2 and a low concentration of oxygen. At the same time, inside the alveoli, through ventilation with air, air is going to have a high concentration of oxygen and a low concentration of carbon dioxide, allowing for that concentration gradient and for gas exchange to occur. So we've talked about some of these, but what are some adaptations of the lungs for gas exchange? Well, we have, first of all, a network of bronchioles, and these lead to alveoli, as you can see here. And this is what increases the surface area. Remember, the surface needs to be large, so that's how this is done. Then alveoli are surrounded by extensive capillary networks with thin walls. As you can see here, they're all over in blue and red. And these are important because that's where gas exchange happens. And then alveoli also have surfactant on them, on the inner part. So you can't really see it on this image, but I tried to point at the inside of the alveoli. And this reduces surface tension and prevents alveoli collapse. So let's talk about ventilation. So ventilation involves muscle contraction. Why? Because it causes air to flow from regions of higher pressure to regions of lower pressure. So during inhalation, the diaphragm and external intercostal muscles are going to contract. And what that's going to cause is that the pressure inside the thorax is going to drop below atmospheric pressure, and thus air comes in naturally, right? That's just through gradients. And then during exhalation, the diaphragm and the external intercostal muscles relax, and the abdominal muscles and internal intercostal muscles contract, causing pressure to rise above atmospheric pressure. So air is forced out. So it's just about trying to regulate the pressure inside the thorax. So when these contract, the others relax, and vice versa. So some measurements of lung volumes is first the tidal volume. So the tidal volume, and this I'm afraid you just have to memorize, but you will be asked to apply this in data-based questions. So the tidal volume is the volume of fresh air that is inhaled or exhaled. Also, I haven't included it here, but the ventilation rate is the number of times that air is drawn in or expelled per minute. So that's important to remember. The vital capacity is the total volume of air that can be exhaled or inhaled after a maximum inhalation or exhalation. That's a key difference with tidal volume. Then the inspiratory reserve volume is the amount of air that can be inhaled after a normal tidal volume inspiration. So that's inhaled after a normal inspiration. So it's not the same as vital capacity because that's a maximum inhalation. And the expiratory reserve volume is the amount of air that can be exhaled after a normal exhalation. So 3 and 4 are opposites. And all of these can be measured with simple apparatus, but you should expect an IB to see graphs and for you to have to calculate this. So bear in mind what each of these are. Okay, so not only lungs are adapted, so that's in humans and in a lot of animals, but plants have leaves, and that's where gas exchange happens. So chloroplasts need a supply of carbon dioxide for photosynthesis, and at the same time, leaves need to avoid losing too much water. So what are the adaptations? Well, there's an outer surface of the leaf, so that's called the epidermis, and it's covered in a waxy cuticle, and that has a very low permeability to gases, so not a lot of gases pass through here. Same here, down here. But that's why there are guard cells, which you can see here, these ones at the tip, and these can open or close, allowing the stomata, which are the pores, to open or close, and the gases to pass selectively. So for example, they will close when there's low water level so that water is not lost, or at night when photosynthesis isn't happening because the sun isn't shining. And then you can't see it here, but also there are normally veins, which are quite big, where the xylem is found, and that's where water is supplied. Then there's also spongy mesophyll, which you can see here, with a network of air spaces. This allows gases to diffuse. And then it's in the spongy mesophyll that photosynthesis can happen, as well as here, the palisade mesophyll. Then we have transpiration. So transpiration is the loss of water vapor from the leaves and stems of plants. Why does this happen? Well, it's because humidity in the air outside is lower than inside the leaf, inside the air spaces of the leaf that we just saw. And therefore, due to gradients, the water will leave the plant naturally. Transpiration rates are affected by two factors mainly. Temperature, so that's a positive correlation. The higher the temperature, the higher the rate of transpiration. And then also by humidity. So basically, the higher the humidity outside, the lower the rate of transpiration. That's because there's less of a gradient, right? And the water doesn't want to leave as much. And guard cells are going to try to prevent as much transpiration as possible by closing when it's not needed to do gas exchange. So that's another type of gas exchange surface. And then this is actually really, really easy. It's just a model density. So you can look at a leaf through a microscope and calculate it, and it's the mean number of stomata divided by the area of field of view in squared millimeters. So just calculate the area and then calculate how many stomata you can see and do the division. Super simple. Okay, so now for HL. Hemoglobin. So this is a bit more complex. So hemoglobin, we've looked at it before. It's a protein found in red blood cells, and it's used to transport oxygen. So if you remember, hemoglobin has 4 subunits. It has a quaternary structure. And each of the 4 subunits in a hemoglobin molecule has a heme group, which you can see here in green. And that acts as a binding site for oxygen. Hemoglobin has a lot of adaptations for oxygen carriage. So binding of oxygen is cooperative. What does that mean? So basically, when oxygen binds to one heme group, it increases the affinity of the other heme groups for oxygen through conformational changes. That also happens in the opposite direction. The second one is its interactions with carbon dioxide, which we'll look at in the next slide. And thirdly is fetal hemoglobin. So that's produced by babies at birth, and it's later replaced by adult hemoglobin. So fetal hemoglobin has a stronger affinity for oxygen than adult hemoglobin. Why? Well, because for a fetus to have oxygen, and its hemoglobin, oxygen has to dissociate from maternal hemoglobin and bind to hemoglobin in fetal blood. That can only happen if the fetal hemoglobin has a higher affinity than the maternal hemoglobin for oxygen. I hope that's clear. That's a mouthful. But yeah, and that happens through structural changes as well. So fetal hemoglobin has a different structure than adult hemoglobin. Okay, so I was talking about the carbon dioxide, the effect on hemoglobin. It's twofold. So firstly, CO2 reduces blood pH, and that reduces the affinity of hemoglobin for oxygen. And then secondly, CO2 can bind to hemoglobin directly, forming carbaminohemoglobin, and that also reduces its affinity for oxygen. This does make sense, right? Why would carbon dioxide decrease the affinity of hemoglobin for oxygen? Well, it's so that in actively respiring tissues, right, where CO2 is being taken out of cells, that reduces its affinity for O2, for oxygen, right? Allowing the oxygen to go into cells to perform respiration. Okay, and finally, dissociation curves. So they look a bit weird. They have an S shape, and basically it's the saturation of hemoglobin with oxygen against the partial pressure of oxygen, so how much oxygen there is. Why this S shape? It's because of cooperative binding, right? As soon as one oxygen binds to one heme, so that's about 25% of saturation, right? Because remember, it can have 4. You see this really marked increase in affinity. Why? It's because of this cooperative binding, right? So there's a structural change, and that means that the other units have a higher affinity for oxygen, so it's much easier for them to bind. And then fetal hemoglobin is shifted to the left, and why is that? It's because it has increased affinity, as we mentioned before. Okay, so let's do some questions to recap. So first, identify X. That should be straightforward. Pause now if you want to think through it. I'll count down to 3. So 3, 2, and 1. It's the bronchioles, right? So X here is at the end is the bronchioles. And then this would be the bronchus, right? And this would be the trachea. Then next question, which is true of fetal hemoglobin? I'll let you think through this one a little more. Pause now. Again, I'm going to count down to 3. 3, 2, and 1. Okay, it's C. Why? Well, remember, fetal hemoglobin has a higher affinity for oxygen because it needs to basically steal the oxygen from maternal hemoglobin. But it's not due to an additional heme group. It has 4 heme groups, the same as adult hemoglobin, but it has structural differences, so that's why. And final question, which muscles contract during exhalation? I'm afraid if you don't remember this, there's no way to figure it out. It's just memorizing. But if you do, then you might not know by now. But again, in 3, 2, and 1, it's the abdominal muscles and internal intercostal muscles. Again, not much to say about this. You just need to memorize this. So remember, the abdominal muscles and intercostal muscles, internal intercostal muscles contract during exhalation, and then during inhalation, the diaphragm and the external intercostal muscles contract, and then vice versa with relaxation, right? So that's it. So hope that was useful. If you have any questions, leave them down below, and I'll see you next week for the next topic. Bye, everyone.